I'm Matt McGee. I'm a grad student here at Davis, and I get to introduce a guy who really doesn't need to be introduced because most of you are quite familiar with him. He's one of the giants of evolutionary biology, pioneered work on adaptive radiation, ecological speciation. And what some of you might not know, he's a professor at UBC, but some time ago, when I was 2, he was a postdoc here at UC Davis. And he's a great guy. He works on a cool system that's given us great insights into evolution, ecology, and now genomics. So here's Dolph.
He said it was going to be short. I thought I was going to be able to relax a little longer. So it's flattering and enjoyable to be here. I've been here, I guess, given a few talks over the years. I did actually come here as a postdoc in 1984 in Tom Shainer's lab. I split a postdoc between Jamie Smith's lab at UBC in Vancouver and Tom Shainer's lab here, and I did a bunch of bird work in the hills to the west of here, and then also at Deep Canyon, one of the California reserves. This was well before my Stickleback years, and I wanted to sort of show you what my trajectory has been since then, since I was here working on birds, and I've shown it downhill, not so much as a statement on the fields that I have tried to learn something about since I was first here. Originally, I worked on problems related to the distribution, abundance of finches, their diversity, the role of character displacement in the structure of species assemblages. I got interested in phenotypic evolution, even quantitative genetics, and more recently I got interested in speciation. But gradually, I'm getting sucked into the field of genetics, and that's partly because so much work is now being done in Stickleback. But I've shown it really as a downhill trajectory because I've gone from a position in which when I started this work, give me an airline ticket, a pair of binoculars, a notebook, and I'm done. I'm good. I can go out into the field, and I was fairly comfortable. Add a computer to that, I could basically do everything by myself. But now, since I've started working in some of these other areas, I've gradually come to be able to do less and less of the area that I'm actually working in. And so I've reached a point of near utter dependency. And I suppose that means that by the time I retire, I'll be able to do nothing at all. And the trajectory is in large part made possible by work that's gone on in the laboratory of David Kingsley and Katie Peichel, who was a postdoc of David's, is now at the Fred Hutchinson and the University of Washington in Seattle. And the two of these have done a lot of amazing things with Stickleback. And I've gotten interested in, helping them, but also applying some of the results that they've been doing. But I did want to sort of ask the question, is this useful? Why am I getting involved in Stickleback genetics and soon Stickleback genomics? Is it even useful? It's really, I guess, the subtitle of the question of my talk. Why go there? Why would someone with sort of a background in ecology now be studying the fates of genes? And so the subtitle really is, or the sub question for my talk is, can we not understand everything we need to know about the evolution of species assemblages with a bit of quantitative genetics and studies of phenotypic evolution without knowing anything about the genes? What does knowledge about the genes actually add to our understanding of this, which is really what we're interested in? Well, maybe, and I want to explore the idea in this talk, maybe, just maybe, knowing something about the molecular basis of variation can help us understand phenotypic evolution, can actually help us to understand the process of natural selection on phenotypes. So that's the thing I want to explore in this talk. I'm going to talk about several projects that we're in the midst of, none of which is complete. An example of where knowledge of the molecular basis of variation has been useful is the idea of a selective sweep. It's actually possible in many cases to detect where selection has acted and had its impact at the genomic level. And it's actually become an industry, but it can be useful if you know something about the influence of those genes on phenotypes. So it could be useful for the more ecologically minded person like myself. But today I want to explore with 3 different projects how we've tried to genetic information to help us understand 3 different things. And the first is I'm going to tell a story about how knowledge of the genetic basis of a trait has helped us, or is helping us, to understand better the mechanisms of selection on phenotypes. The second is a situation in which we've tried to use our understanding of the genetic basis of a trait to understand a little bit the forms of natural selection and its role in maintaining variation in population. And then I want to end with a study in which we're looking a little bit at the genetic signatures of an adaptive peak shift. So the story for Stickleback begins around 13,000 years ago, where in the part of the world where I work, the Pacific Northwest, up until around 12,000 years ago, was covered by ice about a mile high. And the ice melted relatively rapidly over a period of 1,000 years or so. And when it did so, the sea level was about 150 meters higher than it is today. And that's largely the result of the weight of the ice pressing the land into the sea. But with the weight of the ice removed, over a period of another 1,000, 2,000 years, the land rebounded. And as it did so, a series of small lakes was formed along the coast. And in these small lakes, which all look more or less the same like this, there are Stickleback. And those are the populations that I work on. And today what I want to talk about is adaptations associated with the transition from the marine three-spine Stickleback, which is the ancestor of all of the freshwater populations that we work on, and still swims around the sea, and the changes that have occurred in freshwater since those lakes were formed and then colonized from the sea. So I'm not actually going to talk about any of the speciation work. I'm going to focus primarily on the process of adaptation associated with that environmental transition. And that environmental transition happened over and over again. Each small lake was basically colonized independently by marine Stickleback from the sea. And there are a series of changes that took place. And I'm going to focus on 2 changes in this talk. And the first is in armor, particularly the lateral plates, which are a series of bony plates which lie along the lateral line down both sides of the marine ancestral form. So I've illustrated them in red so you can see them fairly easily. And the marine species is built like a tank. It's plated down the sides, it's got long spines, it's a highly defended animal. The other aspect, which I've traced in green, is just an overall changes that have occurred in the shape of the animal. And that involves many kinds of structures, but I'm going to sort of summarize them all together by changes in the position of landmarks around this outline. So I'm going to toggle between these 2 images just to reinforce in your mind the kinds of changes that I'll be talking about. The first thing that you can see from this slide here is that the armor is much reduced. And again, I'm going to focus on the lateral plates. So whereas the marine species might have 30 plates down each side of the body, most freshwater populations have on the order of 5, 6, or 7, and there are many populations, including one that I work on, that has none at all. And the other thing that has changed is the shape. The freshwater form is more deep-bodied. The head is larger, the jaws are rotated downward, the position of the medial fins is pushed up the body. And I'll toggle between them so that you get an idea. The marine form has a very small head. It's an endurance swimmer and is built for minimizing drag. It's very streamlined. And that's because most marine populations are actually anadromous. They migrate out to sea after they're born, and they migrate back into streams where they breed. They may encounter freshwater Stickleback in those streams. But even you could watch thousands of these animals in large schools off Vancouver, and they're just constantly swimming against the tide. They're basically built for endurance. Whereas migratory habitat has been lost in most of these freshwater populations, and they're certainly not built in a streamlined fashion. You can tell by the changes in the shape of the head. So we know something now about the genetic basis of these shifts, much more about armor than shape. The main gene we now know that's responsible for the reduced number of lateral plates down inside of the body is a gene called ectodysplasin, or EDA. It's a gene also known from mammals. Mutations, coding region mutations in this gene in humans produce a condition called anhydrous ectodermal dysplasia, which the afflicted individual has no hair, no teeth, and sweat glands, a variety of deficiencies in dermal and epidermal structures. But in the Stickleback, it's an adaptive mutation, and it's probably not a coding region mutation. And the other trait that I'm going to focus on, as I said, is shape. And there are some QTL for shape that are reasonably large effects, but in the main, it's polygenic. There seem to be lots of genes that influence overall shape. The story of how it was determined that EDA was the gene responsible for the reduction in lateral plates is just a great story. This is a work by Pam Colosimo and others, with help from just about everybody else in the Kingsley lab, that eventually, through brute force, narrowed it down to a region that contained 4 genes, and then in order to show that this particular gene, which was the best candidate within the region, was in fact the gene responsible, is that they did transformations, injected Stickleback with constructs. And the hilarious thing about this experiment, and also moving even, was that they used a mouse construct, because there being no Stickleback construct available. But you know, we're all fish. It worked. But one of the useful things for people like me in the work that's been done is that there are now, of course, very cheap markers that one can use to genotype individuals at this major locus. Another thing that was learned in the course of this study is that in different freshwater populations around the world that are low-plated, it's the same gene every time. So not a single exception has been found now. At the time, there were about 15 populations, but the size, the number of populations that have been sampled has increased, and it's EDA every time. And not only is it the same gene in most cases, but it's the same allele. Or rather, the alleles that are found in different freshwater populations around the world are all related to one another. And this here represents a phylogeny of the low armor alleles shown in blue, and the high armor alleles shown in red. So these are the ones found in marine populations. The blue ones are the ones found in freshwater populations, and with the possible exception of a population in Japan, which nevertheless is low-plated, and the cause is EDA. We're not actually sure whether it's a new mutation. But there's just a big old clade of low alleles. And the clade of low alleles joins the main trunk of the marine high armor allele sometime between 2 and 10 million years ago. So this low armor allele is not just older than the lakes in which I'm studying these populations. It's orders of magnitude older. In fact, I think it's older than any known freshwater stickleback population. And yet freshwater is the only environment in which this allele is favored. How can this be? How is this possible? Well, the answer must be that the Marines brought it with them when they colonized freshwater at the end of the Ice Age. And sure enough, if you go into marine populations today and look for the allele, you'll find that about 1 in 200 individuals is a heterozygote. So it's a carrier of the low armor allele. It's largely, but not completely, dominant. So most of the individuals that carry the allele nevertheless have all or most of their lateral plates. So they just look just like everybody else, more or less. Whereas in freshwater, the low armor allele is fixed. So it's present in the sea even today. So it's not unreasonable to imagine that when populations were colonized at the end of the Ice Age, that they brought this allele with them. It's the best possible explanation for that data that I showed you. And not only that, we suspect that had I been able to prove that the allele is maintained in the sea at low frequency through hybridization between the marine and freshwater populations when they breed in streams together. They often hybridize. So freshwater populations might be the perennial source of the alleles even in marine populations. Every once in a while you find an individual in the sea that is, actually is double low armored. So that's shown in the individual on top. It still looks, it's a marine, but it has both of its copies are the low armor allele. So they do occur. It's present in the sea. Now, the first of the three stories that I want to talk about, and again I want to remind you that these are works in progress. I'm not going to tie up beautifully at the end of each story the answer, because to tell you the truth, we're still scratching our heads in each of them. But I thought that would be more fun than giving you a nice tight donut story, because it will convince you that I still have stuff to do. So the marine population is completely armored, and we're not entirely sure why or what the adaptive significance is of high armor. It's thought that the presence of these lateral plates reduces mortality by birds and fish. And here's an image of a trout that's grabbed a stickleback. So the fact that they have plates doesn't deter predators from trying to catch stickleback, but stickleback are pretty hard to handle. They're pretty spiny. They can actually lock their spines open even after death. We found them in the throats of fish long after the stickleback have died. And as a result of this difficulty, many of them actually escape. And they escape with many fewer lacerations if they are protected by this armor than otherwise. That's one of the hypotheses for why they're out there. Also, stickleback can't really swim. They row. And in the sea, endurance swimming is aided by a stiff body, and it's possible that the plates contribute in that way. It minimizes drag. But I want to focus on the question, why has the low armor allele been favored in freshwater? And a variety of hypotheses has been suggested. One is that, well, there are fewer predators in the sea, certainly fewer bird and fish predators, but there's another difference, and that is in freshwater lakes there are insects. And they're the main killers of certainly of young stickleback anyway. They're eaten by back swimmers, notonectids. They're eaten by dragonfly nymphs. Certainly in the lakes they're no longer endurance swimming, so there's no need to minimize drag to the same extent. And possibly the result of fewer ions in the water may actually be more expensive to produce the bone that make up these plates. When Kerry Marchenko in my lab tested a number of ideas, including the idea that it might actually be linked to the survival in salt water, that the armor, that the ETA allele might actually affect viability in salt versus fresh water, which is an old hypothesis that was published by Hughes in the first issue of Evolution. And so he was able to discount that, but in the process of testing this idea, he showed quite clearly that at least in freshwater, and we think also there is a difference in the sea, but it's smaller, that simply having plates causes you to grow slower. And it's a fairly substantial difference. So what we have on the right-hand side is the average growth rate of families of low armor and high armor marine populations raised either in saltwater and freshwater. Certainly you can see that in freshwater the low armor families grow, the individuals in those families grow much faster. It might be as simple as that, plate building section burden, particularly in freshwater. And there's almost it's not quite a twofold, but it's a fairly substantial growth disadvantage. Kerry also included a freshwater population which is polymorphic at the ETA locus, something I'm going to talk about in that second story. But even in this freshwater population, he showed that the low armor families grow faster, the individuals in low armor families grow faster than the individuals in high armor families. So even in these populations, which have been in freshwater for some time, there is apparently a cost of producing the lateral plates. Now in the lab, this might not mean very much, but as I mentioned, in the wild, in these lakes, insects are everywhere, and they're the main killers, and they hit primarily the small stickleback. And the probability of surviving through that period increases with body size. So basically, it's feasible, it's possible that because the larger you are, the more invulnerable you are to insects, the faster you grow, then the sooner you get out of that incredibly vulnerable range. And there are other reasons why small size might be disadvantageous and a good idea to escape as soon as possible. But that's, we have a lab experiment which suggests that there's a difference in growth between the high armor and low armor phenotypes, but what we thought would be useful is to ask whether that translates in any way to fitness. And so students in my lab did an experiment in which we investigated the effects of different armor phenotypes and specifically different EDA genotypes to survival growth in a wild population or a field experiment. So what they, as in we as in they did was go to Oyster Lagoon, which is a population of Stickleback that actually breeds in the sea. This is an elevated lagoon, which is accessible only at the highest high tides. But it's a population that breeds completely in salt water, and as a result, there's no streams coming in, so there was no chance that we would somehow accidentally catch a freshwater individual through the sampling. And so what they did in this population is they trapped and screened 35,000 animals and brought back a total of about 350 individuals that seemed to lack the full number of lateral plates down inside the body. So as you'd expect, each one of these 35,000 fish and a host of volunteers over a period of about 2 months to collect these individuals. And almost all the individuals are complete, but a fraction of them is partially plated. So most of this work was done by Rowan Barrett and Sean Rogers. So then these fish were brought back to the lab and they were genotyped using these markers, and it was found that about half of them were heterozygous at the EDA gene. So they were carriers of both of the alleles. And so what we did was basically do what happened 10, 12,000 years ago when bodies of fresh water formed and marines colonized. We colonized 4 freshwater ponds. These are about 20-year-old dirt-lined ponds. They're about 23 by 23 meters, 9 feet deep in the center. And they're basically just holes in the ground, but they were used for this experiment. And what we found in each was that the adults that we threw in from the sea, threw them into fresh water, they bred happily and produced offspring in the expected 1:2:1 ratio. So we had a cohort of individuals produced in the pond, and then what we did is we tracked their fates through life until they first bred. So every 6 to 8 weeks, a sample of 50 individuals was taken from each of the ponds and genotyped, and this was repeated basically from birth until first reproduction. And what that involved was using various trapping methods there shown with a seine, basically catching the stickleback, sifting through the contents of the seines, and sampling 50 and then throwing the rest back. And this occurred summer or winter, rain or shine. In one sampling episode, it was necessary to push a boat out into the ice and jump up and down to crack the ice, and then just kind of row around and break the ice up so that we could do the same. We, as in they, could do the same. And the main reason, or the main guide that we had in carrying out this experiment was the idea that there's simply a growth disadvantage to being completely plated, and that the low allele might have an advantage. And so what we expected was, by around the time that the final number of lateral plates is completed during development, there should be a size difference between the different genotypes associated with the different plate morphs. And that from that point on, we should see effects of those size differences on survival. And that we should see, therefore, the frequency of the low arm allele rise as the fish aged up until the time of first reproduction. And so that's what we observed. By October, after the first summer, the fish were introduced in July, I believe, and by October the fish were on the order of 30 millimeters, and they had developed the final plate number that they would have for the rest of their life. Each of these lines refers to the relationship between the body size of the individual and the number of low alleles it possessed. And so this was repeated in October, and that persisted to to some extent by May, by which time these individuals were beginning to reproduce. So there was a positive relationship. Basically, having low armor alleles increased size, and therefore growth. And sure enough, across this period, the frequency of the low alleles shot up. The average advantage to survival across the period from October to May was about 50% in favor of individuals carrying a low allele. So an unbelievably strong selection coefficient. So we saw the expected growth differential, and certainly consistent with that, we found that this translated into a dramatic change in survival. So this is, there's no breeding across this period. This change in frequency is entirely the result of differential mortality. So they're dying like flies. But the completely plated individuals are dying much faster than the low plated individuals. So all this seemed well and good, was consistent with the predictions from this growth hypothesis, but then we saw something which we did not expect. And that's shown here. So when we started the experiment, the frequency of the low allele was 50%. We introduced heterozygotes so that there would be absolutely no variation from replicate to replicate in the starting frequency. And almost immediately upon birth, the frequency of the low allele began to dive. And the selection coefficient that we observed was almost as large as the selection coefficient in the opposite direction that occurred after plates had reached their final number. What we observed basically is that even before they have plates, that there is strong selection on this major gene. Now I told you that I would not have a neat and tidy story, a complete story to tell you for each of the three studies that I was going to tell you about, because I have no idea why there was strong selection in favor of the complete allele. And again, there's no reproduction happening across this period. All of this change in frequency is produced by differential mortality. These fish are dying like flies, and but one group or one genotype is just dying at a much faster rate than the other, until about October, when it reverses and starts screaming in the opposite direction. So while this part of the study was consistent with our expectations, this was a complete surprise to us. Now, at the same time, we made crosses from the same pool of individuals sampled from Oyster Lagoon. We created the genotypes in the lab, and we kept all of these genotypes in both saltwater and freshwater, and found that absolutely no selection like this was observed in the same cohort of animals in a laboratory environment. And so the cause of this strong selection is the result of some interaction between genotypes and the environment. It's an ecological mechanism, but we don't yet know what it is. So just to summarize the results from the first part of the first story so far, the goal of this experiment was to test the mechanism of selection. And a number of the results that we observed in this experiment were consistent with that mechanism. And the growth difference, we see it in a wild setting, and it is associated subsequently with tremendous differences in survival of different genotypes. But we found opposing selection area in life that was similar in magnitude, but in the opposite direction, which diminished the lifetime advantage. And as I mentioned, no selection alleles was seen in the lab control. And one of the lessons that we've learned so far from doing this, related to the main topic of the talk, is that the major gene for lateral plates, discovering the major gene for lateral plates was just a tremendous success. It was a triumph. This is a major change that happens repeatedly over and over. It's subject to strong selection. But now what we've discovered is that this major gene does something else in addition. And it seems clear that we're not going to understand the evolution of lateral plates until we also understand the other impacts of its major gene. And those still work for the future. The second story that I want to tell is a study of selection in a polymorphic stickleback population. So Kennedy Lake is a gigantic lake on Vancouver Island, and there are a few such large lakes in this area of British Columbia that have stickleback populations that are polymorphic. So most freshwater populations, virtually all, are fixed for the low allele. but there's a few populations that are polymorphic for armor. Some individuals are completely plated, some individuals are low plated, and a few individuals are partially plated or intermediate. And Carey thought this would be a great opportunity to look first at whether ETA is the main gene underlying this polymorphism, and then through repeated sampling maybe get an idea of what sorts of selection pressures are acting on this polymorphism by way of providing us with some insights on what factors might maintain this polymorphism. The polymorphism does not appear to be transient. So collections were made in 1963, and we're in our Fish Museum, so Carey took samples in 2004, 2008, and the frequencies of the three different morphs, lateral plate morphs, was about the same. This is Carey Marchico enjoying a tasty barbecued stickleback on the day of his PhD defense. Low armor when I hope. So what Carey found was that, first of all, ETA is associated with this polymorphism and is surely responsible, is the major gene underlying this polymorphism. But much to our surprise, we found that there was a deficit of heterozygotes at this locus. So the bars indicate the observed proportions of the three genotypes at the ETA locus. And then the little horizontal bars indicate the frequencies that you would expect under Hardy-Weinberg expectation. So there's a serious deficiency of heterozygotes, which really surprised us. Because, well, you have to imagine we thought we would try to look at the potential role of selection in maintaining this polymorphism. And actually, selection is very difficult to maintain polymorphism in which there is a heterozygote disadvantage. So I thought one of the first things that occurred to us, well, actually, maybe there's a sort of mate. So potentially, maybe the lows prefer to mate with the lows, and the completes with the completes. No one had ever documented anything like that before. But maybe, if they feed in different areas, they might, choose one another more often when choosing a mate. And so it became important to test this possibility by genotyping embryos. So what Carey and a bunch of others in the lab did, they went out to Kennedy Lake during the breeding season, collected adults right off the breeding ground, and they show the same heterozygote deficit, so it's repeatable generation after generation. And then they genotyped the embryos and found that there was no Hardy-Weinberg deviation whatsoever. So it's completely vanished. There's no non-random mating. There's no sort of mating does not explain the deficit of heterozygotes in the adults because it's gone in the embryos. So we only have one season's data. Carey's going to go out again this year just to confirm that it happens year after year, but certainly the adult deficit in the heterozygotes is repeated year after year. So there's no non-random mating. The only conclusion that we can draw from this is that selection is actually selecting against the heterozygotes. And if you plug the frequencies into a model in which you estimate the selection coefficients necessary to go from this frequency distribution to the one seen in adults and back again, you estimate a selection surface that looks like this, where the heterozygotes are selected against substantially. So I guess this is story number 2 in which what we got was not what we expected. And maybe it's because there are not very many examples yet where people know something about major genes have gone out and looked at the frequencies of different genotypes, but I don't think there are very many examples of single gene polymorphisms that are maintained under heterozygote disadvantage. It's actually very difficult to maintain a polymorphism with heterozygote advantage because when mating is random with respect to genotypes, then whichever homozygote genotype is less common, on average will be producing more offspring that are heterozygous than the more common genotype, and thus will be selected against, and it can be really fast, particularly with selection coefficients like this. So I have to conclude my story by saying I don't actually know yet, I have no direct measurements of selection to confirm that this is the shape of selection, but it does raise the question, well, how is selection maintaining this polymorphism at all. And to my knowledge, anyway, the only thing that I can think of to maintain this polymorphism is that selection must be frequency dependent. So there are some models that Michael Torelli has worked on an ecological model that has this attribute. If it should happen that as one of the homozygotes become very rare, it gains an advantage so that its frequency increases, and it is actually possible to maintain a polymorphism despite the heterozygote disadvantage. So there's only one bit of information that I have that relates to this, and that is the niche differentiation between morphs is one way in which you could plausibly get frequency-dependent natural selection favoring the rare homozygote genotype. So if they feed at different niches, and so to some extent are regulated independently in those different environments, then well, that becomes one way in which a polymorphism like this one could be maintained. And so Carrie, with a group of other people, sampled the different morphs from Kennedy Lakes, and using isotopes of carbon, were able to show that there's actually a significant difference between the two morphs in isotopes of carbon. So this is a method that's commonly used to distinguish individuals whose ultimate source of carbon is either from the littoral zone or from the pelagic zone. And if you look at the species, Stickleback species pairs that I also work on, the limetics are all down here and the benthics are all up there. It's a clear-cut association between the relative abundance of the carbon-13 isotope relative to 12 and the degree of pelagicness of the diet. So all that we know is we don't actually know what they're feeding on that would explain this difference in carbon signature. But they are ecologically different. Plausibly, therefore, there's a mechanism out there that could help to explain this polymorphism, but we don't yet have any direct evidence. So to summarize the second part of the talk, the goal of this project is really to try to understand the role that selection plays in maintaining this polymorphism. And sure enough, it's determined by variation at the eta locus. It's a major gene. But the surprise was that there's a deficit of heterozygotes in adults, but not in the embryos, not explained by assortative mating. And without direct measurements of selection, we still need to obtain direct measurements of the selection. But the observation is that there is disruptive selection. It's the only explanation we're left with this for why heterozygotes are selected against. And then there's an open question that polymorphism might nevertheless be maintained by frequency-dependent selection, but to demonstrate that is still work that remains. So the third story that I wanted to talk about concerns the transition from marine to freshwater in overall body shape, which, as I said, is a polygenic trait. And with Sean Rogers, I've investigated the model of adaptation that we think describes what happened when these lakes formed, marine forms colonized them, and they began to adapt to the characteristics of fresh water. So a sort of a geometric picture of what happened can be seen as an adaptive landscape where you have, two of many traits that might be under selection. The third dimension is fitness, and basically what happens as the population adapts is that it becomes gradually more fit under the conditions of those environments, and then its rate of evolution slows. So I'm going to call that an adaptive peak shift. So we think of it as a kind of a geometric model for how the process of adaptation takes place. And perhaps to make that more tangible, I'll show you some pictures of stickleback evolution as indicated by fossil series that Michael Bell has made. So these are from a mine in Nevada, and they represent very fine time slices of an ancient lake bed that was colonized at some point, we'll call it time zero, a highly armored stickleback form. And through fine sampling of the stickleback that are fossilized in this bed, Michael was able to measure a variety of their characteristics through time and show, for example, the diagram on top shows the number of dorsal spines started out high and then at first changed relatively rapidly, and then slowed, equilibrated to some lesser value, within about 5,000-6,000 years. So this is how I imagine an adaptive peak shift would look if you saw it in the fossil record. And so what would it look like genetically, though, if there was such an adaptive peak shift? So to do this, I'm going to tell you a little bit more about the natural history of shape changes. As I mentioned, the ancestral marine is a very streamlined animal, and the freshwater populations are derived. And if you throw a whole variety of morphological measurements into analysis and calculate the major axis of shape variation among all these populations, what you'll find is that the major axis of shape variation is one in which the marine populations lie at one extreme and all the freshwater populations lie on the same side. So it's a massive, repeated, parallel evolution experiment. Shape always evolves, basically in the same direction, more or less, once the marine populations colonize freshwater. And it's genetic. One of these 4 dots is marines raised in freshwater ponds. So as they spend time in freshwater, they adapt. In terms of their overall body shape, they become more deep-bodied, the head becomes bigger, the big jaw becomes more rotated, and so on. But there's nevertheless quite a large degree of variation among populations today in those shape characteristics. Now, we don't have a fossil record for the adaptive peak shift that we think describes the process of adaptation, but there are some theoretical models for the genetics of adaptation that could help us generate an expectation. And Ronald Fisher was the person who sort of developed this model that Alan Orr has subsequently worked on. And to show you how the model works, I'm illustrating it in 2 dimensions for just 2 traits, trait A and trait B. And the third dimension is fitness. And what we imagine is that a population colonizes a new environment and starts the process of adaptation at some distance away from the optimum in that environment. And I've illustrated it here for just 2 traits, but try to imagine that there are n traits, a fairly large number of such traits. And that as the population adapts, it acquires mutations that eventually bring it closer to the optimum. And that describes the series of adaptive substitutions that that underlie this process of adaptive peak shift. So key to the model is the ancestor starts at some distance away from the fixed optimum. That when mutations occur, they actually affect multiple traits, not just 1 or 2, but they affect many traits. And then by sequential accumulation of these mutations, the population colonizes. So that's a sort of a genetic model for adaptive peak shift. And what Fisher pointed out was that in this case, small effect mutations are more likely to be advantageous than large effect mutations. And that's because no matter how many trait dimensions there are, very small mutations are just as likely to bring you inside the circle, where fitness is higher, as outside the circle, where fitness is lower. So whereas large effect mutations are more likely to take you outside the circle than inside the circle. And this discrepancy becomes greater and greater the greater is the number of traits that are influenced by mutations. The greater is the number of dimensions of this adaptive surface. And what Alan Orr showed was that by taking into account the fact that even though it's true most mutations of small effect are more likely to be advantageous than mutations of large effect, mutations of large effect are more likely to fix than mutations of small effect. And the net result is that when adaptation occurs through the kind of adaptive peak shift, that you should not be too surprised to see the occasional large effect mutation, even though most of them will be small. The overall expectation is that approximately you should end up with an exponential distribution in the effect sizes of genes underlying adaptive peak shift. But the second part of the prediction is that if large effect mutations do contribute, that they should happen while the ancestor is still far away from the optimum. And that's because the closer and closer is the evolving population to the optimum, the more likely it is that a larger effect mutation will take you away from the optimum rather than toward And so that's the sort of the depiction of an adaptive peak shift, and we can contrast that with perhaps a moving optimum, the idea that perhaps lakes at the end of the Ice Age were very different from what they were today, and populations evolved as lakes changed, and that in general they never did climb an adaptive peak, they just trekked an adaptive peak that started out fairly close to where they began. And this leads to contrasting expectations about the distribution of effect sizes. First, you should never see large effect mutations. And secondly, that there should be no effect of where the optimum is today relative to where the ancestor is today in the distribution of effect sizes. So we decided to explore this prediction to see whether it matched the observations that we see in lakes of different types in which these marine populations have adapted. And to do this, we looked at shape, and I'm just showing you this slide to say that shape is really a complicated trait. It's really the x and y coordinates of a series of position traits, and each of these positions represents, where a particular bone occurs, and so on. I'm not going to go into any detail, but the overall advantage of using something like shape is that it is polygenic, and also all the traits that describe shape are in the same units, millimeters. So in some sense, we can lump them all together. And to explore this process of adaptive peak shift and what sort of genetic imprint it might leave, we focused on the shape characteristics of Stickleback in two different lake types. So this is a similar graph to the one I produced before, but now I've identified lakes belonging to two different classes. And the one class of lakes shown in dark are lakes that contain one species of Stickleback and cutthroat trout and no other fish. Whereas there's another class of lakes out there in which the Stickleback populations occur in otherwise very similar lakes that also contain prickly sculpin. Prickly sculpin is an intragilled predator of Stickleback. It eats littoral invertebrates and thus competes with Stickleback, but it also eats Stickleback, and thus is a predator. So it's a competitor that eats its competitor. It does, it interacts in both those ways. But the observation that we make when we sample populations that occur in these two kinds of lakes is that repeatedly the overall shape of Stickleback from a lake with sculpin is about midway between the kinds of overall Stickleback shape that you see in lakes that contain over cutthroat trout from the ancestral state. So if you have only cutthroat trout, generally those populations are much more derived, have evolved much greater distance from the ancestral state than have those populations that occur today with prickly sculpin. So I'm going to assume, for the purposes of our study, that these represent two adaptive peaks. And I'm going to justify that by saying we see this repeatedly, independently. The populations stop here when they occur with sculpin, and they stop here when they occur without sculpin. And so you can see where I'm heading with this. I'm going to try and devise a test using these populations in which I look at the genetic basis of adaptation by an ancestor to a near optimum versus to a far optimum. And the basic idea is that these two lake types represent environments in which the ancestor began at the same place but didn't go as far, or that they adapted to be nearer in the case of the sculpin lakes and in the case of the trout lakes. And what this means is that, in general, because this ancestor starts closer to the adaptive peak, a given large effect mutation is more likely to take you away from the optimum than would be the case if the ancestor was farther away from the peak. So it's to test this second prediction from the geometric model of an adaptive peak shift, that the large effect mutations should be most advantageous, or are most likely to be advantageous and contribute when the ancestor is far from the peak and when it is near. So to do this, I'm only going to be able to show you the results from two populations that we've done so far. So we've got two more waiting. We're nearly there, but the data I'm going to show you just now comes from Cranby Lake, which has only trout, and Pag Lake, which has trout and sculpin, and they were both crossed in the same ancestral form, and then we did all the usual QTL analyses and then estimated the effect size of those QTL on individual traits. And this is just a histogram of the effect sizes influencing shape in those two freshwater populations, one representing the far optimum, the Cranky Lake cross, and the other representing the near optimum with Sculpin present, represented by the Pack Lake cross. So this shows that there is a wider variance in the effect sizes in the far optimum population. None of these effect sizes are so large that they would actually leapfrog over the near optimum. They're not gigantic effect sizes, but they are, in some cases, large. And there is a difference between these two distributions, but as I said, this is only one comparison, and I'll believe it when we see the same, if we see the same pattern again in the remaining two populations that we're still working on. So to summarize the third part, our goal in this study was to detect the genetic signature of an adaptive peep shape. And what we found so far is that the genetics of shape match the expectations, that the frequency of large effect mutations seems to be greater when the population, when we looked at the population that in which the ancestor had adapted to what we conclude to be a farther optimum. We've also done work to show that the distribution of effect sizes is approximately exponential, but I didn't show you that. There are probably lots of things which influence whether large effect mutations or not contributes to the process of adaptation in general, but the distance to the adaptive optimum might be one of those factors. So I just want to mention a few caveats before I wrap up, and that is that we're looking at QTL, not individual mutations. So it's always possible that what's a large QTL is actually two or more mutations whose effects are sum. And it'll be a while before we can figure out which of those is correct. So genome sequencing would be necessary before we can ever do that. Secondly, the theory actually refers to the effect size of mutations on fitness, the distribution of effect sizes on fitness, not on traits. But the OR has shown that more or less the predictions are the same for traits as for fitness if you can assume that large effects in traits also are proportional to effects on fitness. And that's an assumption that we are in the process of testing, but I have not yet shown you yet. And then finally, replication is another thing we need to do. We have a couple more lakes being investigated, so we'll be able to report on that soon. Next time. So is knowing something about the genes useful? Why should a person like myself who works on evolutionary ecology of adaptation and speciation really go out of his way to learn something about genetics? And I'm not sure I have a complete answer to that question, but what I've been trying to do is to explore the idea that knowing something about genetics can actually be useful, not just because it's a superb story in and of itself, part of the natural history of adaptation in this particular group, but also that it might tell us something about phenotypic evolution and the process of natural selection itself. So these three stories were meant to sort of address that point, and I'll leave it to you as to whether we have learned anything substantial enough to justify yet all of the time and expense needed to do genetics on a non-model organism. In this case here, it's easy to say that it would have been impossible to detect selection without the gene. And there's a couple of examples of that. In the pond experiment that I began with, we had strong selection on the major gene underlying lateral plates before lateral plates developed. And so, at least in that practical sense, having a marker for lateral plates before they develop is a useful tool for documenting phenotypic evolution. And in the case of the Kennedy Lake polymorphism, which I didn't show in detail, the Kennedy Lake polymorphism is such that there are actually very few partially plated fish in the population. Most heterozygotes are actually completely plated. We can't identify them yet from their phenotype, but they clearly seem to have a difference in fitness. So the gene is definitely something that allows us to dig a little deeper, and I think we'll be better able, if we succeed, we'll be better able to understand the maintenance of the phenotypic polymorphism by doing something about the dynamics of the gene. And that would probably involve the effects of this gene on other traits, which we haven't yet determined. So what I've been exploring is the idea that studying the effects of selection on genes is a useful complement also to gene hunting. It was a triumph in my mind that the Kingsley lab identified this gene responsible for the polymorphism when we thought, and that was back in the times when we had a reasonably good idea what we thought the adaptation was. Lateral plates are reduced in fresh water because there's lower predation and maybe lower ion. But now we've discovered that actually the major gene governing lateral plates does something else. And we're not going to understand lateral plate evolution without it. We're not going to be able to do this for every gene in the genome. I'm aiming for gene number 2 so that we can now do the entire series of studies again using PIDX1 or KIT ligand. It's not truly practical, but what I imagine, or certainly something we should consider as possible, is that we'll learn some general lessons just from studying a field. But again, I'm not sure. Anyway, that's my interim report on the work we've been doing at the genetics of adaptation in Stickleback. Thank you for listening.
So if I understand correctly, the notion for the first story, the early failure to survive and the later success of the mutant individual, presumably has something to do with a pleiotropic effect of the gene. Yeah. And so have you been searching for such things as disease resistance as a possible? I mean, the epidermis, after all, is a major barrier to parasites.
No, that's a great point.
Is that sort of work?
So it's work that we hope to do. We have not yet begun that. The other possibility we've considered is that might influence behavior, maybe expose them to predation or something like that. So,
do you know whether the sampling is, or does the sampling need to be perfect? In other words, that your sample of the little guys is a true reflection of what's actually there in the pond?
Yeah, it's a good question also. So we went through a lot of hand-wringing over the question of whether this trend was the result either of some genotyping error, a seasonal effect of genotyping, which just gets worse in the fall and improves in the spring. But we checked with other markers, and so we thought, well, are we biasing the way that we're catching these animals if there is an effect of the gene on behavior? And so we tried catching these things in every which way: traps, seines, dip nets, underwater. You name it, we tried everything, and we found no effect of trapping method. It doesn't completely rule out, but I think we've given it as good a shot as we possibly can. I think that the changes in frequency are real.
Are, in that last bit there, are those fish, are the sticklebacks in sculpin lakes, are they ecologically intermediate also? Are they sort of more open water, more lignetic?
So we haven't done enough field studies of the ecology. We've only recently discovered that they are different in their phenotype. They kind of looked similar, but it shows that you don't really see it until you get it under the microscope and measure it. So we're not entirely sure. I'd like to go to these lakes and do some stable isotopes and see if they're ecologically intermediate as well. They do seem to spend more time offshore feeding on zooplankton than in the other single species lakes. So I think they are more zooplanktivorous. Whether they continue to use the littoral zone as well, I'm not yet sure.
What other stickleback traits would you especially like to identify genes for?
So in general, one of the projects, Katie Peichel was here not long ago and mentioned a collaborative project that we're doing, in which we're going to try to test some of these ideas concerning the contribution of genetics to the maintenance of sympatric species in the presence of gene flow. And so we'd like, for example, to know whether reproductive isolation in the genetics of reproductive isolation in these sympatric species of stickleback conforms to, one of the ways that Felsenstein said you could avoid the problem of selection recombination antagonism that happens when there's gene flow. And one of them is that that the traits that are under selection and the preferences for the traits and the targets of those preferences all map to the same region. So one of the projects that we have going now is to try to look at the genetics of reproductive isolation between the limnetics and benthics. And at the same time, we're looking at a variety of morphological and behavioral traits that contribute to their differences. So, and that phase of the study will identify mostly just QTL, but in the meantime, David Kingsley is sequencing the genomes of the parents of our limnetic benthic crosses. And so hopefully we'll be able to merge the data sets and have some idea what those genes might be if we can actually find genes for traits like reproductive isolation. So that's still pretty hairy and uncertain whether that will work, but give it a shot. So speciation genes would be something I'd really like to know more about. And not just because, wow, it'd be nice to know what the speciation genes are, but because we'd like to test some of these models for how species can form or how reproductive isolation can become strengthened even when there's gene flow, which is what we think happens.
Yeah, so this primary axis in which these stickleback are evolving, how much of the variation is off of that?
Yeah, I was afraid someone was going to ask me that. So the majority of variation is in that one direction, but I can't remember how much is off. So most populations have some idiosyncrasies as well.
And then when you were estimating the step sizes in the two different populations, how does that compare to, you said it's not like clearly going the whole way, but is it going some very, very small step compared to the distance they have to go?
So the largest effect mutations that we found actually, cover half the distance.
And the small ones are?
Cover half the distance of the far one, say. So perhaps the near peak could have been reached in a single mutation and that one been favored, but it wasn't favored. But again, the provider.
Those are in, I guess I didn't quite get the units on it. Those were in units.
They're all in millimeters.
Along that.
They're all in millimeters.
But your axis is in something more complicated.
So the shape axis that we used are the units are actually millimeters of change in position of the landmarks in the x and y coordinates. From one as you change from one pitch to the other. So they're just ordinary traits in that sense.
I guess the assumption underlying that is that the shapes that you regard as being adaptive because they are what is represented in the contemporary populations with trout or sculpin, the genes underlying those shape changes are those that responded directly to selection from the sculpin and the trout, whereas I suppose it could have been, say, genes underlying behavioral changes that allow them to persist, live in a somewhat different way logically, and that shape change followed after that. Okay.
So what you have here is what is controversially known as a natural experiment, which is to say no experiment at all. It's an observational study, and our main goal was really to ask whether we could use the expectations of the theory to guide our interpretations of what's happening. But even when we finally settle down on knowing what the mutations are, we're still going to be left with different, maybe the lakes are different in some other way. So there's still, it's an observational study, but one in which we knew that when we did it, and did it anyway. Because I think the theory has to be useful. It's not going to help us explain, even relatively simple patterns like this. So I thought it was worth a try for that reason.
Back to the study number 1. Would it be useful to know, or even practical to do, to know what are the causes of mortality at the different stages of the, in other words, supposedly caged,
yeah, so we brought them in the lab and they just didn't die, so we have to somehow, so it's hard to actually figure out.
It might be impractical.
But we could do things like, remove all the insects from one pond or something. Actually, that's impractical too, but we could certainly make a stab at it. We could reduce mortality from insects.
Do you change the water temperature in the lab to represent a change in temperature?
We attempted that as much as possible, that's right. So we did this in an environment chamber. The only thing we were not able to do was to bring the environment chamber temperatures as low as that which occurred in the well. And it was one of these cases of brand new environment chambers supposed to be able to reproduce them, but when we actually did it, it did not happen. So that never happens here.
Did you bring the predators into the lab?
No, we haven't done that. So that would be, on a small scale with juveniles, we may be able to look at survival of the different genotypes in the presence of the predators.
All right, maybe one more question.
So, since we're asking about the Kennedy Lake story, in terms of the explanations for why you have the maintenance of heterozygotes, you don't have a genome, but do you know anything about the linkage of that gene to other genes? So,
Yeah,
physical linkage?
That's a really good question. So the question of what it's linked to actually influences all of the stories that I've just described. So in the first story, we're just taking heterozygotes out of the sea, and we don't know how long or how much of the rest of the freshwater, allele might be attached. The ETA gene was discovered by using linkage disequilibrium mapping in a polymorphic California population, which is thought to have been undergone something like 10,000 generations of recombination. And what they found was that the chunk of DNA that contained the ETA locus still contained 4 genes. So even after 10,000 years, there were still 4 genes in a block. And so they were left with the question, which one? And that was one of the motivations for the experiment. I suspect that the block is larger than that, even in most of these populations. Kennedy Lake is actually younger than 10,000 years. So I would bet that there are other linked genes, and how to remember the name of them. One of those genes has been implicated in some other organism in salt handling. Another one has been implicated in parasite resistance. So the problem exists that some of the effects that we're seeing here are the result not of ETA, but of linked genes. So I'm hoping that we can eventually tease that apart.